Okay, we are good to go. So our next speaker, which is the last of today, is Massimiliano Rossi. He is currently a staff bioinformatician scientist at Illumina. He obtained his PhD in University of Verona and collaborated also with University at Helsinki with both Professor Veli Makinen and Sami Pugliese. After that, he did a postdoc at University of Florida under the supervision of Professor Christina Boucher. Uh, his work has mostly been involved with string and string indices, but today he will talk about Dragon, which is a multi-genome mapper and a pangenome reference. So thank you, Massimiliano, for being here, and please, the stage is yours.
Thank you very much for the very kind introduction and for having me here. So yes, so hope that everyone is enjoying this pangenome biohacking, which I really think is a very nice initiative. And yes, I'm a staff bioinformatics scientist at Illumina, and I'm going to talk about Dragon, the multi-genome mapper and pangenome reference, focusing on the quest for accuracy gain in those dark regions of the genome. So let me first start talking a little bit about what Dragon is, and then I'm going to talk about the concept of the multi-genome mapping that Dragon introduced 4 years ago, and that has shown great accuracy improvements over the years. And finally, I'm going to talk about how to build and use pangenome reference in Dragon. So for example, from the HPRC assemblies that are really great resource. So what exactly is Dragon? So Dragon is a dynamic read analysis for genomics. It's a suite of industry-level hardware-accelerated bioinformatics pipeline that are used for secondary analysis. So where Dragon places itself in the sample to answer pipeline, then Dragon is right after the sequencer primary base calling and before the tertiary analysis and reporting. So basically, Dragon pipeline start from raw reads, and it produces variant calls or counts depending on the application. And the output of Dragon is then fed into the tertiary analysis, where the genetic data is then interpreted. And Dragon has been designed to be our most accurate, comprehensive, and efficient secondary analysis pipeline to date. It has been designed to maximize genomic discovery. So it is really accurate. It produces accurate results, achieving 99.89% accuracy score using the Precision FDA Truth Challenge V2 benchmarking data, empowering the second generation multi-genome technology and the machine learning, driving unprecedented accuracy. It's really comprehensive. So as I said, it's a suite of bioinformatic tools, and it can analyze whole genomes, exome, methylomes, and transcriptomes with a single solution that replaces roughly 30 open source tools. And it's really efficient. So as I said, it's hardware accelerated. It can process a 40x whole genome sequencing in roughly 34 minutes with all supported callers. What does it mean? It means that it can make small variant calls, structural variant calls, copy number variant calls, and other specialized caller that are included into Dragon. And all these callers gives a really comprehensive view of the genomic variants of the analyzed sample. And is also efficient in the sense that it can reduce the memory footprint of the raw reads up to 5 times with the Dragon Aura compression. So it's again a really comprehensive suite, and it has been widely used over the years. And as of December 2024, 8.5 million human whole genome equivalent samples has been processed through Dragon. The human whole genome equivalent is essentially 120 gigabases of raw and aligned read for a 30x genome. And we can proudly say that Dragon has been cited more than 1,500 publications in total, still as of December 2024. So in October 2024, there has been quite a nice achievement for Dragon that has been published in a paper in Nature Biotech, where we describe the Dragon workflow in the germline pipeline of the context of Dragon. Dragon has also other pipelines like somatic, RNA, and others, as I said. But in this paper, we focus on the germline pipeline, and we show the accuracy and the comprehensiveness of Dragon on the genome in a bottle samples, and as well as we reanalyzed the whole 1,000 Genome Project cohort that is also available on is online. The results of this reanalysis of the 3,202 whole genome segments. So let me describe briefly the Dragon germline workflow, just to give you an idea of what it does and why we say it's comprehensive. So essentially, the first step, we said Dragon takes an input row reads. So the first step is to align those reads against a reference genome. And we get the help of population haplotypes to better guide the alignment of these reads to a given reference genome. And we report this alignment into a reference genome coordinates, which could be either GRCH38 or 37 or the new T2T CHM13 reference. And these aligned reads could be saved into a BAM or CRAM format, but they can be streamed directly into all the variant callers that Dragon offer. So like, for example, once aligned, we can feed these reads into the small variant caller, which essentially takes these reads, identifies active regions. It also assemble plausible haplotypes and determine the per read likelihood. And then we go through a genotyping phase, and the results of this genotype generates candidate variant that then goes through a machine learning step that evaluates those candidate variants and makes the variant calls. Another caller that is the structural variant caller that can call deletion, duplication, insertion, inversion, and breakhands, and which first identifies breakpoints and then generates local assemblies for each of these breakpoints, and then perform SV scorings and genotyping for those variants and emit the variant calls. There is copy number variant caller that calls duplication and deletion of larger than 1,000 base pairs that goes through steps of binning, shifting level segmentation, and then adjustment of the copy number variant codes using also this structural variant caller informations, which really improves the CNV detection accuracy. And then we also have a bunch of targeted callers. Those are callers that are dedicated to individual genes. So why? Because these genes are really important, for example, for pharmacogenomics or for genetic diseases. And those are usually in difficult to map regions, so those dark regions of the genome that are difficult to map or are into segmental duplication, so that they have homologous regions where the reads is difficult to align the reads correctly. And they can undergo also recombination events like gene conversions that could make the genotyping of those genes really challenging. And another caller is the STR caller, which calls tandem makes calls into tandem repeat regions, and it leverages catalog to identify those regions, collects the reads, and realign those reads into the repeats, so they then can be genotyped, and the call can be made. And finally, there is also iterative GVCF genotyper workflow that essentially now this is something that works at cohort level. So for example, for the 1000 Genome Project, we can collect all the variant calls made by Dragon, and then we can batch them and process them, aggregate into cohort level genotypes, and then generate a multi-sample VCF file for this cohort, which again is what we did in this paper. And I talked a lot about all the methods that are available, and you basically can get all these variant calls in 30 minutes of computation time from the raw reads, alignment, and all the variant calls that I explained you right now. So it's really efficient, and it's also really accurate, as we are going to see. But I promised to talk about the multi-genome mapping and the pan-genome reference. So let's now dive into a bit of the history of the multi-genome mapping and in Dragon over the years. So here I have a table where that describes the evolution of four major components of Dragon. So the multi-genome mapper and the pan-genome reference, as well as machine learning and the native alt handling. So by multi-genome mapper, what we mean is the method that's used to map and align reads to the reference. And for pan-genome reference, what we refer to is the collection of samples that are used to augment the reference to improve mapping accuracy. So the multi-genome mapper and the first generation of the multi-genome mapper and the pan-genome reference was introduced in the version 3.7 of Dragon. And the first pan-genome reference was composed of 16 samples of European ancestry. So the pan-genome reference augmentation, what they do enable the mapper to select the standard reference contig alignment more accurately and to better estimate the mapping accuracy for those alignments using the population information provided by the pan-genome reference. And it is important to notice that the variant calling is largely unaware of the pan-genome reference. Uh, why? Because all the reads alignment are reported in the reference coordinates, not in the pan-genome coordinates. But the variant caller benefits from the improved mapping accuracy. So the better we can place the reads, the easier is the job for the variant caller to make the right calls. So the whole system appears like a known pan-genome analysis externally, with the pan-genome reference serving as an internal method for enhanced accuracy. And then in version 3.9, also machine learning was introduced to improve the variant calling accuracy. And in the version 3.10, the pan-genome augmentation was also extended to include additional haplotypes from the MHC regions to improve the accuracy on those on this important region as well. And in version 4.2, the pan-genome reference this time was expanded to 32 samples now. And we also expanded the ancestry of those samples from European to a global ancestry sample. So we tried to cover more and more genetic diversity. And finally, in the 4.3 Dragon, which is the latest release, is the second generation of the multi-genome mapper was introduced and developed. And this enabled the expansion of the pangenome reference to 128 samples. And this expansion is really shedding more light into the difficult to map regions of the genome. So how all this history translate itself into variant calling accuracy. So here I'm reporting the same history of Dragon across versions into accuracy on small variants, so SNPs and indels, on the AG002 samples from the genome in a bottle with the 4.2.1 truth set. So as you can see here, the different major improvements that I talked you before are reflected kind of one-to-one into the accuracy of variant calling accuracy. So the first major improvement is the multi-genome mapper with the 16 sample pangenome reference. So this is basically comparing using a pangenome or using a versus using a linear reference. And you can see that's like a 45% error reduction when you use the pangenome reference. And then the second major improvement was the machine learning, which played a very nice role by with introducing like a 33% error reduction in version 3.9 compared to the previous version. And the last major improvement, as I said, is the second generation multi-genome mapper with the 128 samples pangenome reference that played another 40% error reduction compared to the 32 sample pangenome reference and first generation multi-genome mapper. So overall, if you compare the latest result on the latest pangenome reference to the place where we start in 3.6 from this graph, this plot here, we have an overall 83% error reduction. So the pan-genome reference and the second generation multi-genome mapper is really helping in reducing the error and improving the accuracy. And this is setting new standards for, so this new version of Dragon is setting new standards for accuracy. So here I'm showing the similar overview on the accuracy over different Dragon versions. And I'm this time I'm showing it with the recent draft of the T2TQ100 truth set for the AG002 on still on a small variant, so SNPs and small indels. And I'm also placing this the Dragon accuracy into context with the publicly available known giraffe deep variant pipeline that runs on the Human Pangenome Reference Consortium assemblies. So what we can see here is that the same milestones applies, the same major improvements have the same effect on the accuracy reductions here, and the error reduction that you can see from the 3.6 version to the 4.3 version is a 42.8% reduction in accuracy. And when we compare to the publicly available pipeline, we do see a 31.5% error reduction when compared to the latest version of Dragon. So here, just to clarify, the pangenomes that are underneath are different because the giraffe D variant runs on the HPRC pangenome, while the Dragon has its own pangenome reference. So we expanded the benchmarking to the whole genome in a bottle dataset. Why we did that? Because we wanted to see how this new mapper plays a role in with different ancestries. So we are covering different ancestries here. So how well the second generation mapper and the 128 samples pangenome reference generalized to the different ancestries. And what we see is that we have 30% on average accuracy improvement on both SNPs and indels with when we compare to the Dragon 4.2, so the previous version of Dragon, with the 32 sample pangenome reference. And with a bit more focus on SNPs, so the accuracy on SNPs is more improved compared to indels. And we do see an even larger, a 41% false negative reduction compared to the previous version of Dragon. So we do correctly detect more variants. And if we compare to the publicly available tool, then the gap is slightly larger, and it goes up to 61% accuracy improvement on average. But at the beginning of the talk, I promised a quest for accuracy gains in the difficult regions of the genome. So those are defined by the genome in a bottle as regions that have other homologous regions in the reference genome for a given read length, number of mismatches, or a number of indels, as well as segmental duplication regions. So as you can imagine, those regions are particularly challenging since it will be difficult for the mapper to distinguish one region of the region of origins of a read based on a very small handful of differences. So here is where the pangenome comes to help. So in these regions, very few difference between the regions can make a very big difference in the alignment confidence. So the closer the aligner can find an haplotype that better match the sample read, the more confident it can be that, on placing this read correctly, on the placement of this read, and consequently it increases the map queue. So what happens is the aligner is essentially uh, make it more easier for the variant caller to make the correct call because it's more confident on placing the read correctly. So when we look at the whole genome in a bottle data set, so again on AG1 through 7, and we focus on these difficult to map regions of the genome, then the impact of the second generation mapper it's higher than on the overall other regions. And we do see like a 37% average accuracy improvement compared to the previous version of Dragon, and roughly a 50% false negative reduction. So it's it's really helping making the right calls in those difficult to map regions of the genome. And so let's now try to look a bit at how the first generation mapper works to try to understand where is this, why is this helping this much. So the first step when to use this the multi-genome mapper is to identify these population haplotypes. And what we do is we select these population haplotypes in difficult to map regions of the genome from phased SNPs and indel calls from the genome reference samples. And since we want to have this haplotype as much accurate as possible, we do filter out low confidence calls and low populational frequency calls, since this could not be as much reliable into guiding the mapping process. And then we do augment the reference genome with in two ways. So the first one is that unphased SNPs are encoded as multi-base codes. And this helps in guiding the alignment, as well as the phased SNPs and indels are represented as multi-genome alternate sequences with unknown leftover to the primary assembly. So this is important. And let's look at this picture on the right. trying to explain why the leftover information is important. So let's say that we are trying to align this read here, and this read has three possible alignments on these three different regions, A, B, and C, where these vertical bars are representing mismatches. So what happened in a traditional read mapping context, then the read mapper would pick the best one, the best alignment, regardless on the relation of each of these regions, because it's completely unaware of that. And the map Q confidence, so the confidence of the alignment will be given by looking at these three regions independently. Now, the multi-genome mapper has the extra knowledge of leftover relationships. So let's say that this region C is actually a multi-genome alt-contig with a leftover relationship to the region A. So essentially, those are encoding different haplotypes, but on the same region. So essentially, what the multi-genome mapper can do is now uh, group group these alignments based on leftover relationships. So it can now focus on picking the best per group alignment and compare only those. So so in this case, it would compare this alignment with this alignment only, and would disregard the 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 alignment to the primary assembly because it has more mismatches than the alignment to the multi-genome alt-contig. And where this is going to help? Well, this is going to help because now we can the confidence score would be computed only on the best alignment in each leftover group. And so this is the first thing. So the MapQ would be improved and the confidence would be higher. And the second advantage is that we can then report the alignment only for each leftover group only on the primary assembly. So only the primary assembly alignment is reported. And this is what is making the variant calling unaware of the pangenome, because the variant calling will just need to look at primary assemblies, and it would benefit from the improved mapper accuracy because the the multi-genome mapper did most of the work in placing the read in the right place. Okay, then moving to the second generation multi-genome mapper. So the second generation multi-genome mapper was designed to scale the first generation mapper both vertically and horizontally. So the vertical expansion basically is necessary to include more and more samples from the pan genome. So in the 4.3 release, we scaled from 32 samples, so 64 haplotypes, to 128 samples, so 256 haplotypes. And here I'm reporting in the right the ancestries of those samples. So what does it mean? It means that including more samples, we include more and more genetic diversity, which will bring more and more genetic diversity into these difficult to map regions of the genome, which will help the mapper to make the right choice on where to place correctly the reads. And then the second expansion is the horizontal expansion. So with this the second generation multi-genome mapper, we were able to expand, not only focus on the difficult to map regions, but expand to the whole genome coverage of of the genomes from those samples. So and what was designed was proprietary optimized data structure that allows efficient scaling when adding more haplotypes, and is also able to exploit long-range phasing information when in alignment scoring. So this new data structure partially replaces the leftover mechanisms of the first generation, but is now dedicated. That's still there, is still useful, but it's now dedicated to more complex variants. And when we released Dragon, we also offer this the pangenome reference on the four major human reference genomes that are available. So AG38, AG19, CHM, the T2T CHM13 reference, and the GRCH37. So we spent quite a bit of time talking about the multi-genome mapper, but there is the other side of the coin, which is the pangenome reference that is really important as well, and those are heavily intertwined. So the mapper and the pangenome reference. So what happened if I want a different distribution of ancestries in my pangenome reference? Like this is what comes with the Dragon. This is the default hash table. But what if we want a different distribution of ancestries? So we can now talk a bit about how to build your own pangenome reference for Dragon. So we are witnessing a tremendous effort from pangenome consortia to generate high quality, high number, and highly diverse human assemblies to be used as pangenome references. So we have the pioneering effort from the Human Pangenome Reference Consortium that's taking the lead with the first release of 44 high-quality assemblies, and has been closely followed by other consortia like the Chinese Pangenome Consortium, the Arab Pangenome Reference, and Pacific Ancestry Pangenome Reference, just to mention a few of them. And all those efforts are calling for methods to leverage the genetic information to improve variant calling and to ameliorate the so-called reference bias. So we started thinking about how could we enable the use of Dragon together with custom pangenome references that are not necessarily one of those specified in these slides, but it could be a combination of those, or it could be an in-house made pangenome reference. So what we did is we developed two cloud apps that allow you to do this. So you can bring your own human assemblies and build a Dragon reference, for example, for a population-specific study, and with the objective of reducing ancestry bias. So the user can combine also their sample with other pre-built assemblies that we provide. So when you bring your own human assembly, then you can process them with the Dragon Pangenome VCF Builder app, which is this first app that basically generates phased variant calls with respect to a given reference genome. And then once a set of single sample calls are generated, then you can use this second app, the Multi-Genome Reference Builder app, to essentially create a Dragon hash table with the samples that you processed and selected. And so the process of making the variant codes from the single sample takes roughly 3 hours per sample. While building a multi-genome reference, it takes roughly 2 hours once all the samples are processed. And everything is available on the cloud. So the reason why we broke down this step in two parts is because now a user could process its whole cohort. And then once its whole cohort is processed, then it can build more than one pangenome reference for Dragon and perform different experiments with different for different population specific studies, for example. And what we did is we made available a pre-built set of samples for the from the Human Pangenome Reference Consortium assemblies, and those can be easily used and selected. So we also made a web interface where the user can select, like for example, in this case, we want samples from the Human Pangenome Reference Consortium. with an African superpopulation ancestry, but belonging to the subpopulation of Gambian, Menden, and Asan. And then we can click the run button, and it will generate a pangenome after 2 hours that can be used. So this is available on BaseSpace and ICA platform from Illumina. And the this could really empower users to do their own population-specific studies using the accuracy of Dragon with their custom pangenome references. So what we did is we did build a Dragon reference pangenome from the 44 samples of the HPRC, and we compared it to the linear reference using Dragon 4.2, the default Dragon 4.2 pangenome reference, and the default Dragon 4.3 pangenome reference. So what we can see is that with this method that we developed, the accuracy that we obtain when running Dragon on a custom pangenome, because this is not as highly optimized as the released Dragon pangenome, but we can obtain similar performance, which is slightly worse than the 4.3 default Dragon hash table results, but it is better than the Dragon 4.2 default results. So you can see that the accuracy of Dragon is really comparable with the one using the default hash table. And overall, comparing to the linear reference, we can see that there is a really large, nice improvement on the accuracy. Also, when using custom pangenome, for example, like the HPRC one. So that's all for my talk. And I also wanted to mention that with the Nature publication, now we mentioned that we do, if you want to try out Dragon 4.2, there are availabilities for academic licenses, and yeah, that's it.
Thank you very much, Massimiliano, for the nice talk. Um, we may have a few questions from
Gianluca ha fatto praticamente la stessa domanda.
Um, okay, yes, I see. We have a question from Gianluca. He's asking, if I understand correctly, Dragon computes a pangenome by adding custom assemblies on top of pre-built pangenome. For example, the one of the HPRC. Can it build a pangenome from scratch without a pre-built pangenome?
Yes. So okay. So here we do provide pre-built assemblies for the HPRC, but there is no need to use them. So if you have a collection of samples, of human samples, you can process them, and then you can build the pangenome without including any of the HPRC assemblies. So the answer is yes. It could be completely unrelated to the HPRC set of samples.
Okay, it's typing. In the meantime, I think few people are typing for question, but still no question yet. So, okay, no, Gianluca replied, how many genomes can you assemble?
So we okay. We need assembled genome to start with. So the input for this Dragon pangenome VCF builder app is assembled genomes. And so as of today, we can handle up to 256 samples in building the multi-genome reference.
Okay. We have another question from the audience, from Luca Denti. He's asking if in the pangenome, uh, are the input genome encoded in the graph as P or W lines, or as a GBWT separated from the graph topology in your output pangenome?
Okay. The mm, the encoding of the haplotypes is proprietary data structure, so it's a different data structure from the encoding that are publicly available.
And this is from me. I suppose there is a description on this data structure, right? Or is it completely private and not uh, usable outside of Dragon?
So it's so as of today, it's not usable outside of Dragon. It's a proprietary of Dragon.
Okay. There might be a follow-up from Luca. In the meantime, I would like to ask um, about the impact. Yeah, sorry, we have this delay between the session and the live streaming. So, okay, no, okay. His follow-up question was my question. So, okay, yeah, we have this a little delay between what we see in this meeting and the live chat, live streaming. So sorry for that. So my question is, how large is the impact of this hardware acceleration? And some follow-up on this, which is, could similar results be achieved with a general purpose CPU? And um, I assume the answer is no, but is there any plans to make these specific hardware open source so that it can be uh, re reused by other entities other than Illumina?
Okay. Okay. The impact of the hardware acceleration is on runtime. Okay. The align the all the hardware accelerated algorithms could be also implemented into CPU mode. That those are algorithms, so the implementation is it could be ported either in hardware or not. And so the impact is on runtime. Again, as I said, you can align and buy and call at 30x genome in 30 minutes, essentially. So that's the main benefit. And about the open source of the code, the whole code is proprietary. So it's is not going to be open source.
Okay, yeah. Follow up for this. Yes, I understand that any hardware acceleration is just acceleration and it can be done on CPU. My point is that without that, I don't know if you did some experiment of that, but is the benefit so high that in practice it would be unfeasible to do it without it?
Mm. Uh, I don't have experiments on this, and I um, I don't believe it's unfeasible.
Mm-hmm.
But I have no idea what could be the runtime that that it would take. It's it should be feasible, but
Okay.
I don't know.
Okay. Okay. Thank you. I don't think we have any other question? You answered my own. Uh, well, actually, why not? This is more of curiosity. Like you have in all these slides for research use only, not for use in diagnostic procedures. So,
Mhm.
um, what is actually missing for be used in diagnostic?
Oh, it needs to go to formal approval
But
to like IVD type of things. So there is a whole formal process that software and methods needs to go through to get approval for dia diagnostics.
so it's mostly like the bureaucratic part, but the
Yeah.